First, I want to thank the organizers for this precious opportunity. Today, I will talk about identification of ethnicity-specific associations in multi-ethnic genome-wide association studies. This is joint work with my student, Yingying. Large-scale biobank studies, such as the UK Biobank study, provide us with unprecedented opportunities to decipher genetic architectures of complex diseases in minority populations. One way to call ethnic-specific associations is to partition the biobank into different ethnic groups according to self-reported ethnicity. However, biobank studies usually consist of admixture individuals whose genomes have originated from multiple ancestries. For GWAS of an admixture population, classical approaches try to adjust for the population structure using either principal component analysis or linear mixed models. However, these methods only claim whether a genetic variant is associated with a phenotype or not, but they do not call ethnicity-specific associations. They do not detect variants that are associated with some but not all ethnicities, and they ignore the effect size differences across ethnicities. Meanwhile, the famous PSD model is a probabilistic model that is able to call the ancestry composition for each individual. There is literature that couples the PSD model with likelihood ratio test to test the association between genetic variants and one binary phenotype. However, PSD model that can adjust for multiple confounding factors and that are scalable to biobank scale GWAS are still lacking. In this talk, we propose a method called MESA that can simultaneously estimate ancestry proportions for each individual and infer the ethnicity-specific effect sizes. MESA is scalable. Its time complexity is linear in the number of ancestry K, linear in the number of SNPs G, linear in the number of cohort size N, and cubic to the number of phenotypes. It reduces the memory consumption from terabytes to megabytes. Therefore, it is applicable to GWAS with hundreds of thousands of individuals and hundreds of thousands of SNPs. Here is a model for GWAS that measures G SNPs for N individuals that originated from K ancestries. The probability for the latent ancestry for the G SNPs of the I's individual is K's ancestry equals to QKI. Then given the latent ancestry for the G SNP of the I's individual, then the observed genotype XGI will follow a binomial distribution whose success probability depends on the K's ancestry as well as the phenotype. Let's look at the data generating process. For each individual, he or she has a person-specific ancestry proportion QI, which will generate a latent ancestry for each SNP of that individual. Then given the G SNP's latent ancestry is the K's ancestry, then the major allele frequency for the I's individual will further depend on the baseline latent major allele frequency for the K's ancestry, as well as the phenotype. Here, we are not saying the phenotype is causing the genotype. We just use the logistic regression to link the phenotype with the genotype so that we can test the association via the gamma GK. Given the model, we can use the EM algorithm to conduct a statistical inference. To speed up the computation, we use stochastic updates with the momentum updates. Specifically, we partition all the SNPs into J random chunks. And at the Tth iteration, we only update the parameters related to the SNPs in a given subset in the mth step of the EM algorithm using Newton's method. With the updated parameters, we will estimate a noisy ancestry proportions based on the SNPs within that subset. And finally, we will update the ancestry proportion using momentum updates, which is a weighted average of the ancestry proportion from the last iteration, as well as a noisy estimation from a current iteration. We use a Bayesian information criteria to determine the number of ethnicities, and we parallelize the computation for the parameters for different SNPs. Because we want to do the hypothesis testing, we need to obtain the standard errors for parameters. Here we use the OX estimator to calculate the observed data Fisher information matrix from the Q function in the EM algorithm. Specifically, we need to invert the matrix in the bottom line. Here is a toy example to illustrate how these matrices look like. When the number of SNPs is large and the cohort size is large, the B matrix can be relatively dense, which makes the inversion very computationally intense. We use block matrix inversion techniques and provide two versions of approximation. The first one is the shrinkage-based approximation. It shrinks elements of the block diagonal of Ox estimators to zeros. And the second one is based on the finite expansion of Newman series. And both approximation provides very good results in practice. Here's a simulation setting. We only compare fast LMM with MESA in the first setting because fast LMM can only detect the association with one phenotype. And for the fifth setting, with 10,000 individuals and 10,000 of SNPs, even with only one rather than two phenotypes, the fast LMM needs to consume more than 700 gigabytes of memory, which is enormous. In contrast, MESA can consume only 16.4 gigabytes of RAM. Here is the assumed effect sizes, as well as the estimate effect sizes in one simulation setting. We can see that MESA can recover the ethnicity-specific effect sizes very well. And the overall computation time, as well as the computation time for the standard errors, are indeed linear in the number of cohort size. We applied MESA to the page study, where individuals are labeled as Black, Japanese, Latino, Hawaiian, and others. After quality control, we have 9,000 individuals measured at over 600,000 SNPs. We focus on two phenotypes, the BMI and the gender. And MESA is able to finish the computation within an hour and a half and use only 6.2 gigabytes of RAM, so it can be run on a routine laptop. MESA correctly identified there exists 4 ethnicities in the population, and its inferred ancestry resemble the self-reported ancestries, but we also provide the ancestry compositions of different ethnic groups for each individual. MESA controls inflation of the p-values very well, and it is able to call ethnicity-specific associations. In summary, MESA can simultaneously infer ancestry proportions for each individual and estimate ancestry-specific effect sizes. It is statistically rigorous as we provide the analytic form of the standard errors, and it is computationally scalable with a time complexity that is linear in the number of SNPs and linear in the number of cohort size. And it achieves over 10,000-fold of saving in the memory consumption. We provide our software on the GitHub, and we acknowledge our funding support. Thank you very much for your attention.